Hello, everyone. We will be getting started momentarily, so bear with us as we wait for a few more people to join the meeting. We're very happy that you're here. All right, we're going to get started today. Thank you so much for joining us. Welcome to Workshop Wednesday. We're glad to have you all here to talk about how to effectively communicate your science to fellow scientists. Before we start today's conversation with our panelists, I want to briefly introduce the people and the work that goes on behind this workshop, as well as the entire Workshop Wednesday series, as well as provide you with the general flow of today's event. So today's workshop has been organized by GSA's Career Development Subcommittee. Roz, Mel, and I have put together the blueprint of this event, but every member of the committee showcased here has been instrumental to make this event happen. Also, a special thanks to our engagement staff from the Early Career Leadership Program for providing us with essential technical coordination to our planning. The Career Development Subcommittee helps build relationships with professionals and companies in the larger scientific community to create deeper connections with the society and its members, and to showcase the versatility of career paths open to all of you. The aim of today's workshop is to highlight the experiences of scientists who have experience in communicating their science effectively through a variety of platforms. So here's the outline for today's workshop. We're going to have our introductions of our two panelists. We're going to hear from Dr. Allie Rosenberg about how to make and present a clear scientific talk. And then we'll hear from Rachel Fairbank on tips for a successful elevator pitch with a sample lightning talk by our own Dr. Roz. So during both of their presentations, please feel free to put questions into the chat. And then at the end of both of their presentations, we'll have a general Q&A. And last but not least, I want to share a few notes and ground rules with everyone in attendance today. All the views and opinions that are shared in today's workshop are those of the panelists themselves and are not the organizations that they represent or have worked for previously. In addition, any mention of a company by a panelist is not an endorsement for that given company or brand. This workshop is a safe space, so we welcome you to ask honest questions to our panelists by dropping them into the chat box to everyone. However, if you're being disrespectful in this place, we will ask you to leave the workshop, so please be mindful of others. Members of our community will be collecting these questions from the chat box, and if they're not addressed in today's discussion, we will try to provide some of the answers after the workshop in the form of some career resources. So you don't need to be writing down notes or resources frantically. This will be recorded and placed on YouTube, and all of the resources mentioned will be collected and compiled by our team and sent to all participants after the workshop. And just so that you know a little bit about who is in attendance or who will be watching the recordings later, nearly everyone is a postdoc or a graduate student, and almost everyone is here because they want to improve their communication skills. And many of you use a variety of methods to communicate your science, from writing and data visualization, long talks, short talks, and education outreach. So I hope you'll find something useful from today's presentations. All right. So I'm going to ask our two panelists to introduce themselves. I have Allie's slide here. So if you want to start by introducing yourself.
Hey everybody, I'm Allie Rosenberg and my pronouns are she and her. As you can see, I've started my own consultancies where I'm going to help scientists with their scientific talks, writing, and their careers. Just to tell you the nutshell of how I got here, I went to a small liberal arts college. Then I worked as a lab technician to figure out whether I liked working as a scientist full time. I then went to graduate school. I then did a 2-year-long postdoc, and I left from there to go work as a medical writer. I left that job to start my own biotech company. After that, I worked at a small biotech for a couple of years, and here we are today. And I'm really excited to give you all a primer on what I think is truly a difficult skill, which is giving a scientific talk.
Awesome. Thank you, Dr. Rosenberg. And Rachel, Fairbank is our other panelist. If you'd like to introduce yourself.
Hi, everyone. I'm Rachel Fairbanks. So I am a freelance science writer and editor with a background in genetics. Just to give you a little bit about me. I got a bachelor's in science in biology with a concentration in genetics and development from Cornell University. Completed the coursework for a PhD in developmental biology at Baylor College of Medicine. And then I went and got an MFA in creative writing at the University of Houston. Generally speaking, I write about science for a general audience. That said, I remember that transition from working in genetics to writing for a general audience very, very well. So over the years, I've learned a lot of lessons about, okay, what can we do if we need to make our science a little bit more accessible? So.
Excellent, thank you. And so we're going to hear from Dr. Rosenberg first about how to make and present a clear scientific talk. So take it away, Allie.
Thank you so much, Molly. All right, so hopefully you all are seeing my screen. Can I get a thumbs up, Molly?
Yeah.
Great, thank you so much. Well, I'm really excited to talk to you all today about how to make and present a clear scientific talk. Just to tell you a little bit more about myself, my training is as a cellular and molecular biologist. My favorite model organism is the zebrafish. I have a foundation in genetics, neuroscience, microbiology, and regenerative and developmental biology. I'm currently in training to become a certified master gardener, and I've got a lot of hobbies, and they include gardening, hiking, trail maintenance, tree advocacy, and I make glass artwork. So why should you listen to me? Well, I've got the training. I did my PhD at the University of Pennsylvania. I did a postdoctoral fellowship and an additional SciComm fellowship at Duke University. I've given a lot of invited talks at numerous national and international conferences. I worked as a medical writer. That meant, among other things, that I made posters and presentations for other doctors and researchers, which featured their own pharma-funded research. And a lot of people that I've worked with before have gone on to win best speaker prizes at the conferences that they've presented at. And when they've given job talks, they've been offered those jobs. So I think you're all here because you probably already think that you should try to give a great scientific talk. But if you hadn't gotten there yet, let me give you a couple of reasons. You might win a prize. It's fun to win a prize, and sometimes those prizes come with actual money. You might become better known in your field and someone that people will seek out as an authority. You might get invitations for future opportunities. So every time I've given a talk at a conference, people have come up to me afterwards with hoping that I would come interview for jobs with them. Future collaborators have come my way. And you might get invited to give a talk at some fantastic tropical location in the future, who knows? Lastly, I think of every talk as an opportunity. The research that you're doing someday, you're going to want to write up into a manuscript. And every time you give a talk, you have the opportunity to craft your argument and figure out what is going to resonate with people so that when you get to writing that manuscript, you kind of have an idea of what your field kind of agrees with you on. So it's a way to test out different times, slightly different ways to say things. So if nothing else, I want you to take 3 things away from this presentation. I want you to practice your talk and stay within the time limits. If you show data, you need to explain it. And every single data slide you show should have a specific title and/or a conclusion statement. So a lot of talks that I've enjoyed present an outline of the topics that they're going to go through. And I think this is a really nice mechanism to bring your audience in and explain to them where you're going to be going. It's almost kind of comforting to know what you're going to learn about in a given talk. And so today I'm going to tell you about the architecture of a well-designed scientific talk. We'll talk about effective visuals and about your attitude and how to be memorable. So I like to think of a well-designed scientific talk as an hourglass. That's the shape of your talk if you can visualize it. So you're going to start really broadly, then you're going to talk about the specifics. That's where your own data fits in. And then you're going to pull back out again and speak broadly about your field. So let's go through that bit by bit. You're going to start with broad questions in your field. Then you're going to give minimal background that explains your field and the system that you work in. From there, you start to tighten it. You're going to ask more specific questions that you're going to be addressing in the talk that you're giving that day. And then we get into the narrows. So this is your own data and any additional background that might be necessary to help explain it to your audience. Then we broaden back out again. You're going to start showing some conclusions that tie your data together. You're then going to zoom out even further and tell us about what your conclusions explain about the larger system or field you work in. You'll finish out with future directions, speaking both in near and long term. And very important, your acknowledgements. I want to go through this now in a little bit of detail and explain a little bit more about what to do in these different phases of the talk. So when you're setting the stage, you want to tailor this initial part to the audience that you're speaking to. You're going to want to ask questions that really draw the audience in. Tell us why your field is interesting, what are the open questions, and really hammer in the rationale for why it's important that you're even giving us this talk and exploring these questions. While you're continuing to set the stage, you're going to give the audience some minimal background to explain your field and system. And I really want to hammer on the idea of minimal background. The question you should have in your mind is how little background is needed for your audience to understand your data. And I say that because your audience can get saturated. You're going to be presenting very complicated information. And maybe you're at a conference and you're coming after 10 other speakers, or there's 10 speakers after you. Your audience is tired. They've been using all their brain glucose on paying attention. Help them out and don't drown them in information. Tell them what they need to know. And so I want you to ask yourself, as you're putting your talk together, maybe even beforehand, who is my audience at this particular talk? And what are they most interested in hearing about? So if you're speaking to a group of neuroscientists, maybe you want to mention things that are specifically interesting to them. Talk about brains and neurons. If it's a general cell biology audience, for example, maybe you're going to focus your talk more towards cell-cell interactions, and the neurons themselves are less important for them to know about. And the way I like to structure my talks is to think about, what does my audience need to know for the next 5 minutes? And that's all I'm going to share with them. And if they need to know more later, I'll share that information later. So as we're getting into the specifics of your talk, you're then going to ask specific questions that the data you show on this day is going to be able to address. So we then get into sort of a repetitive structure that you'll use. You're going to show your data and give intermediate conclusions in the middle of your talk. So you'll ask a specific question. You'll show the most relevant data. That doesn't mean you show your entire figure. Maybe you just show a couple of panels that really help get to the heart of what you want the audience to learn. And then you're going to give a specific conclusion about what that data shows. Then you ask the next specific question and repeat the process. And like I said, if the audience needs to know more background, this is where you can present a little bit more in this repetitive structure. Now, this is the section that you're going to expand or contract depending on how long your talk is. So if you have a much longer talk, a 45-minute long seminar, you'll do this loop several times. If you're giving a 12 or 15-minute talk, maybe just 2 to 3 times is all you'll get through. So you'll really highlight the most important data. And this is the section, again, here's where you trim or expand. Your rationale and background are so critical to getting the audience on board with you so that they understand your talk. That's not the place to take away from. So there's a quote from Professor Susan McConnell. She's at Stanford University that I love. Present just enough data to be convincing, but not so much that the audience is flooded with information. And I do want to put a plug in for Dr. McConnell. She gives a truly excellent talk on how to give a scientific talk. It is on YouTube. I've put the link in here and I'll put it up again at the end of this. And these slides will be shared so that you all have these slides as a resource. And I really encourage you to watch her talk and adopt her practices. If you can do that, you will give fantastic talks, or at least you'll be well on your way. So moving on, you've introduced everything, you've talked about your data, and now you're zooming back out again. You want to give a cohesive ending to your story, tie your data together in a satisfying way, maybe present a model at this point. And then zooming out even further, what can you tell us about the big picture that your conclusions have something to do with? And then lastly, those future directions. This is the section that if you're giving a job talk, you'd probably give a lot more information in. And it's really important that you do this because it can show the audience that there's more to learn, that you're interested and excited to do it, or that someone else in your field should probably pick it up and do it. Lastly, acknowledgements. I think this is probably the most important part of your talk. We all know that science is a group effort, and it's important that you acknowledge everyone else that you've worked with. So if you have data, figures, or cartoons that are generated by your lab mates or collaborators, make sure you thank them. Anyone that provided you materials, some of which examples I've listed here, make sure you thank them. And then throughout your talk on your slides, you should be referencing the literature that you might be speaking to establish your background or otherwise. And if you've borrowed slides or cartoons from other collaborators, you should also put their names up on that slide. And you'll see that throughout on my own slides. So an important thing you need to do is figure out how long your talk is while you're making it. And I wish I could tell you how many slides you should be showing. Unfortunately, because of movies and animations, that's kind of impossible. So you need to practice speaking out loud while timing yourself. I would recommend doing this multiple times and train yourself to speak slowly and clearly, as if you're in front of your audience. It's the only way to properly time things out. So when I do this, I sort of edit at the same time because that's the first chance I get to really hear what my words sound like as they're coming out, if the sentences make sense next to each other. So I go through multiple rounds of talking to myself, editing, re-saying that slide until I get it to a place that I'm happy. And if you're having trouble figuring out what you want to say, write a transcript, write yourself some speaker notes. I do this all the time, especially for really short talks where I don't want to have to think about what the slide is going to be when it comes up. And I just want to be able to hit all the right beats. So take advantage of speaker notes, especially if you're on Zoom. You could have a second screen or a printout right next to you if you need to. And then after you've practiced by yourself, you'll want to practice with friends or colleagues, and I'll talk more about this later. So in summary for this part of the talk, I want you to start broadly, then present background and data as needed, and then end broadly. And you should be asking yourself as you put this together, who's your audience? Can you hook them with a topic or a link to some kind of topic that they're interested in hearing about? And what background do they actually need to hear about? And then practice out loud with the timer to determine your speech length and edit from there. All right, so moving on to effective visuals. You should use colors that can be seen easily by anyone. So think about being colorblind friendly. This image to the bottom left has a singly labeled red neuron. Our colorblind friends would not be able to see that, unfortunately. So something you can do is pseudo color your images, usually in one of the Adobe suites or otherwise, where you can recolor that red to a magenta. It'll show up better on projectors and most people will be able to see that. Now, if you're only working with one channel in microscopy, I would recommend just change it to black and white. It actually provides the best contrast. It'll be easiest to see. And if you're showing micrographs, I think you should use a black background. Now, continuing on with color theory here. I don't know if you all can automatically see everything on the left. It's kind of hard to look at. It actually hurts my eyes to see the yellow on the blue, the red especially on the blue. I hate looking at. I really recommend keeping things simple. Use a black background with white or yellow text. That's the most friendly way to look at a lot of data, especially if it's a micrograph. Otherwise, use a white background with a black or dark blue text. Now, as for fonts, I recommend size 18 to 48. You don't really need to go bigger than that. And a really good tip is to look at your screen from 6 feet away. So let's do a quick little exercise. If you don't mind, get up and walk away from your computer. Walk about 6 to 10 feet away and take a look at the screen. You should be trying to see from here if you can see the graphics that you're showing and if you can see the fonts that you need to talk about. And if you can't read or see any of that easily, it's too small. You got to rejigger that slide, make it bigger. And I only use fonts as small as 14 point for citations on my screen, nothing else. So here are some top tips I want you to think about for graphics and visuals. And I'll go through these in examples. You should be showing one piece of data or one idea per slide. You should be explaining all of your data if you show it, and you should have a specific conclusion that is demonstrated by that data. If you show figures, think about how you can crop or redraw them to highlight pertinent information and remove what's extraneous or distracting. And also think about how you can use plain language labels or color coding to help guide your audience. So you should show one idea or piece of information per slide. And I'm not doing that on this slide. There's a lovely figure to the left that I've pulled from a paper. And I think this figure is really beautiful. And you're probably not listening to me because there's too much on this slide. You don't know if I'm going to talk about the micrographs. You don't know if I'm going to talk about one of the graphs or schematics below. There's too much here. If I was going to show this, I would take it apart and show it panel by panel and probably split this into at least 3 different slides. So you can still talk through all the same things, but break it up. This is another figure that I think will just confuse your audience. And I can't tell you the number of talks I've been to where a 800-piece signaling pathway was shown. Your audience will see this and they will just turn their brains off. You've lost them. Showing something that's complex for the sake of demonstrating complexity doesn't do anything for your audience. The thing you want to hold in your mind is, how can I make things clearer to help tell my story? So instead of showing this, show this. What you should be doing is simplifying, redrawing, removing anything you're not going to talk about, and highlighting what you will. So just minimal, think minimal. You should also explain and label things in simple terms. So this is a slide that I made a long time ago. I'm going to walk you through why I like this slide. So we're starting out with a title, and in this case, it's a question, and it's going to clue the audience into what I want them to look at in the piece of data I'm going to show. So I asked, how do macrophages interact with injured nerves? I then put in these color-coded labels that match the colors of the cells in the micrograph. And I didn't put in here that this is a MINX1 GFP transgenic line, for example, because that's not actually important to my audience. My audience doesn't really care about what genes are being tagged here, but they do need to know what kind of cells I'm looking at because that's important. So I simplified the labels and color-coded them so it's easy to understand. I put in information on timescale. This was a movie I was showing, so I wanted them to understand in real time how these cells were interacting with each other. And then at the bottom, citations and credits. So where can you find this information? And who gave me the transgenic line that I very nicely got to use? I am a huge fan of animation. I don't know if you guys have quite picked up on that yet, but I really think it can help you with your talks. And the thing it does is that it slows your audience down so they follow your speech instead of reading ahead. If they read ahead, they're not listening to you and you've lost them. So there's a couple little tricks that I want to teach you. One is that, of course, you can use the animation pane in PowerPoint. Another thing that's really helpful is just duplicating your slides. And each slide has the next part of your animation. In that way, you're kind of not stacking lots of little lines and figures and things on top of each other. And it makes it a little bit easier to see and to manipulate. So that's there to help you out. Another thing I do is for automated video play. So if you're showing video information, what I like to do is have 2 identical slides. The first slide just shows a still frame of the video. And you can then use that time to orient the audience, explain to them what the data is, what they're going to be seeing, what to look for, and then you just hit next. That second slide is set to autoplay your movie. That way you're not figuring out with your cursor or whatever how to find the play button during your talk. So this is a slide that I made, and even when I look at it, I recoil. There's too much here. So here's what it looks like when I can animate it in. So I have the opportunity when this is animated to go bit by bit and explain to the audience what my system is, what manipulation I'm going to do so they understand the experiment. And then I'm explaining how I'm scoring the regeneration of these axons so that they understand what I'm doing with my data later. So I can walk through and explain to them what weak regeneration looks like, what moderate regeneration looks like, strong regeneration, and then how that all comes together into a graph. So on my y-axis, I show the percentage of nerves that regenerated within one of these three categories. Whoops, didn't mean to hit forward. And then on the bottom, I'm showing what the genotype is. Now, I'll show this slide, and what I'll do next is show a pretty data-heavy slide. So let's walk through this one as well. So again, my title is going to explain my data in the system that I'm testing. So the title is Regeneration is Impaired Without Schwann cells. That's the conclusion. And I'm talking about 4 different kinds of mutants that I'm working in. That's my system, right? Then I'm going to point out here that I just explained this scoring system right on the previous slide. So the audience is primed and they know to expect more data that looks like this. My graph axes are labeled just like before. And then I'm going to explain everything on this graph. So if I was talking through this slide, I would say on the first 4 bars of this graph, you'll see that I have, this represents 4 different wild type siblings for the mutants that I'm working with. And most of the axons that have regenerated in these animals are in the strong category of regeneration. Now, in contrast, the last 4 bars are our mutant animals. And what you can see is that most of the nerves have regenerated in the weak category. What this would suggest is that this isn't an issue of a missing gene, but actually a missing cell type, which is what is shared between these 4 different kinds of mutants. Thus, we can conclude that Schwann cells are required to provide directionality to axon extension. If I wasn't going to talk about all these different mutants, I would delete the different bars on this graph. I wouldn't show more data than I'm going to talk about because it's confusing and your audience isn't actually going to listen to you. They're going to be trying to figure out what you didn't talk about. So lastly, and I know I'm going fast, there's so much to talk about and giving a great talk, and I wish I had more time to speak to you all, but let's take a few minutes to talk about attitude and being memorable. So give practice talks. They are so essential to becoming a good speaker. I already talked to you about practicing to yourself. The next thing you should do is practice with coworkers and friends. And if people are nice enough to listen to you and give you feedback, make sure you're kind about how you receive that feedback. It is hard to receive feedback and it means that you have to do a lot more work. That's tough, but I want you to think about accepting that feedback gracefully and gratefully because that is a gift that your friends and coworkers are giving you. Don't argue with them. They're trying to help you. And just know that if someone didn't understand your point, it's because you were unclear. It means you need to rethink how you're presenting that data or how you're speaking about it. And if those people are incredibly nice, maybe they will help you figure out how to say it. If not, that's really your job. And then rework your talk. Take that advice, rework the talk, and repeat this process, especially if they're kind enough to do it a second time. That's a sign of some awesome friends or coworkers. The next thing I want to tell you is to present often, present to varied audiences. There's so much good that can come out of that. And I'll just blaze through that. And so let's talk about attitude and presence. How can you become a dynamic speaker in person and while you're on Zoom, now that we're doing that all the time? So I want you to smile and speak loud and speak slowly and try to modulate your voice. That part is hard for me to explain, but hopefully you've been hearing me actually change the cadence of my speech and how loud and my pitch as I'm talking. If you are lucky enough to present in person, use a laser pointer. It is the best tool that we've got to really help focus your audience's attention on something specific so they can follow you really exactly. If you're Zooming, you should stand up. You can't see me. I meant to take a photo of myself, but I have my computer propped up. I have a lamp in front of me so that my lighting is decent. And I tested my audio last night by Zooming with a friend who listened to my practice talk. And lastly, let's talk about the question and answer period. I think the question and answer period is one of the most important parts of your talk. It might seem scary. It's not scary. It's useful. And the thing I want you to think about is that the audience is on the same team as you. Everyone there wants you to give a great talk, and everyone there, if they're asking you a question, they're genuinely interested, and that's like another little gift they might give you. So smile. And I find it really helpful to repeat the question back to the audience member that asked it. You want to verify that you actually understand their question. Often they come out in kind of a confusing way. So you're allowed to restate it in a way that makes sense to you. This gives you a few extra seconds to think of the answer. And it also helps the audience out in case someone didn't hear the question, which is super common. And if you have a friend or colleague in the audience, have them take notes for you. If you're anything like me, you are going to be high on adrenaline. And if someone suggests a great paper you should follow up with, or someone to collaborate with, or a great experiment, that stuff flies right out of my head. So have someone help you out. So with that, I would like to thank a bunch of people. I would like to thank especially my thesis advisor, Mikhail Granado. Mikhail signed me up for every speaker series that existed. I'm positive of that in graduate school. At the time, it felt like a lot of work, but it really taught me how to give a speech and to all different audiences. So I really have to thank him for that, and for the entire lab for all of the work that we all did together to perfect each other's talks. I want to thank Professor Susan McConnell. I've actually never seen her speak, but she has a talk up on YouTube. It's linked here again, and it is fabulous, and you all should watch it and do as she says. I want to thank the GSA and the Career Development Subcommittee, and especially Molly Maddie, who has been a great collaborator over the years and has done a lot of work behind the scenes to put this together. So with that, I will just leave you again with some best practices. And thank you all so much for your attention. Very happy to be here and to help you all out however I can. So thanks.
Excellent. Thank you so much. And one other reason why you should give a talk is so you can get little emoji applauses. The world's currency of appreciation and validation these days. So thank you so much, Dr. Rosenberg. Look at all these applauses coming in. Wow. So if you all have questions for Dr. Rosenberg on how to give an effective talk, please put them into the chat and we will get to them at the end when we do a combined Q&A for both of our sections. I'm going to start resharing my screen so that we can get to our next speaker, who's going to be Rachel Fairbank. And let's do that.
Thank you for upgrading my title, Molly. Okay, so I am giving a talk. I am going to give you a short presentation on an elevator pitch, and then we're going to hear an example of an elevator pitch, where I will offer feedback. Now, an elevator pitch incorporates all of the advice that Dr. Rosenberg just walked us through, but in a very, very greatly condensed fashion. And you also probably won't have visuals. But, you know, an elevator pitch in its essence is what do you do when you have 1 to 2, maybe 3 minutes at the most to answer the question, what do you do? You know, what does your research do? And I think of an elevator pitch as a bar exam. You know, if you are in a crowded bar with tons and tons of distraction, there's noise, people have their phones, their friends are here, it's a Friday evening, they're tired. Can you explain what you do in a way that is memorable and that leads to that person saying, oh, can you tell me more? That in its essence is an example of what a successful elevator pitch is. And again, like it's very, very condensed. So you really, really have to be very, very careful and intentional about what details you are offering, as well as how you are framing your elevator pitch. Next slide, Molly. Okay, so I have 3 short principles that I always keep in mind when I am trying to explain what I do. Number 1, people understand and remember far less than we think. And that is going to be true even if it is a scientist that works in your exact field. They could be distracted. They could be worried about something. They could, be, tired. And all of that, like it leads to them maybe missing out of field details, or maybe not paying quite as much attention as they realize. You could also use a term that they don't understand, and it will confuse them long enough that they've lost their train of thought, and you've lost them. So remember, like, people understand and remember less than you think. We are also all really guilty of being embarrassed to admit that we don't understand. Like, I've lost track of the number of seminars that I've sat in and not understood a thing, and I was just too embarrassed to admit, like, hey, I don't know that term, or hey, I got distracted and tuned out in the middle of the talk. And if people do get confused and they ask you a question that shows that they're confused, that is an act of courage. And it's an act of courage that you should be grateful for. So, since people understand and remember less than you realize, you want to start and end with the big picture. Why are you doing this research? What is the impact going to be? It goes back to that hourglass that Dr. Rosenberg showed us. And in this case, that hourglass is really, really condensed. It's short. So the biggest thing that you want to focus on is, why are you doing this research and what might the impact be? Like, that is what you want people to remember. And again, getting back to this idea of starting and ending with the big picture. Every single detail that you add in the elevator pitch needs to reinforce the big impact. It is easy to get sidetracked on tangent. It's easy to get lost in the details. You have, as scientists, we have the instinct to explain everything. We don't want to leave out all of these precious details. But if you only have a couple minutes and you are in an environment where people are distracted or overwhelmed or have other things going on, you absolutely have to cut out every single detail that isn't even remotely relevant to what you are trying to do. So keep it short, keep it simple, and make it as memorable as possible. Like, you will know your job. You will know that you have done your job if 1 or 2 weeks later, someone says, oh, hey, Rachel, she works on understanding Alzheimer's or something like that. Like, if they can remember one detail 1 to 2 weeks after you've given your elevator pitch, you have done your job. So fewer details, but make them count. Okay, with that, I'm going to turn it over to Raz, who's going to give us an example.
So, hi, my name is Raz, and I will give an example of an elevator pitch, and then we can discuss it. So my name is Raz, and I will tell you about my research. This is my actual research that I work on. So I work on a gene called Jumanji 1.2, who's a positive regulator of the mitochondrial UPR, which is a machinery in the cell that is in charge of taking care of proteostasis in the cell. Now, what I was interested in is in seeing whether if I activate this mitochondrial UPR using Jumanji overexpression in cephalic glia cells in the nematode C. elegans is able to confer positive effects on the worm. And what I found is that activating this response is able to prolong lifespan of these worms and also protect them against stress. So different stresses that we can give to the worms. Then I wanted to see what are the genetic requirements for this communication. So I tested a bunch of mutants. So I tested ATFS1, which is a transcription factor in the mito UPR, DVU1, another transcription factor, Lin65, a chromatin regulator. And I saw that the communication that the cephalic sheath glia do with the rest of the body is dependent on these genes. And additionally, I continued on to actually try and study on the cellular level what happens and whether these glia cells are communicating to specific cells in the body. And I found that these cells are able to send a signal to intestinal tissues and muscle tissues. What I'm currently trying to do is try to figure out what is the secreted model that is going between these two tissues. And that's it. Thank you.
Thank you so much, Raz. Okay, so let's start talking about applying a few of these principles to this elevator pitch. And I'm just gonna walk Raz through some of what he does and in relation to some of these suggestions, so that you can start seeing, okay, this is maybe how I can think about it in terms of explaining my own work. So number one, I am going to start by saying, listening to that, I don't remember nearly as much as I would want to. And I also don't understand nearly as much as I want to. Now, I spent about 7 years working in a C. elegans lab. They did have a project on aging, still confusing. So, and a lot of that is not necessarily that I'm not capable of understanding, but when someone is speaking and they're going, someone is speaking, it's hard to keep up. And then once you fall a little bit behind, it becomes even harder to keep up. So, you know, remember that. People understand and remember less than you think. So you do have to be very, very careful with the number of details that you're introducing. So, okay, now let's get into the big picture. Yes, I do want to say, like, I did ask Roz I did ask Roz to make a few mistakes. But yes, let's start getting into big picture and impact. So Roz, in your opinion, what is the really big picture of your research?
So I usually, when I want to actually present this, I start very broad, as you both suggested. And I actually talk about the ability of the nervous system to control aging of an organism, which I think is a very interesting concept. And then I go and focus on distinguishing between neurons and glia, since my project focuses on the glial part. And yeah, I think this is like the major point I try to like emphasize.
Okay. And see, here's the thing. So I did not ask to hear Roz's elevator pitch before this seminar because I wanted to hear it fresh right now. Now I picked up on the aging part, and that's definitely something interesting, but I did not pick up on the nervous system and aging connection. And that's actually really interesting. I like that a lot. We're all interested in aging. You know, it is part of being human, you know. So that's something that if you start off with, I am interested in understanding how the nervous system affects aging, that is something that's going to hook in a lot of people. It's going to hook in people that are also C. elegans researchers that maybe also work on a different aspect of aging. It's also going to hook in just, you know, your average person who's like, why am I aging? You know, what's going on when I start, you know, when I start feeling my age? So, you know, that in writing, we call that the hook. So that in the sense that it hooks you, and brings you into the story. So that's the hook. Now, given Roz's presentation, I missed that hook. And I'm really, really sad that I missed that hook because it is such a great one. So yeah, start and end with the big picture. And in this case, the big picture is I'm trying to understand the connection between the nervous system and aging. How does the nervous system affect aging? And if you start with that and you end with that, people are going to remember that. And Roz will have done his job if, again, like a couple weeks down the line, someone, says, oh, hey, Roz, I met him. Like, what does he do again? And someone says, oh, yeah, he's looking at the, how the nervous system affects aging. That's one short, very simple sentence. But if you can keep that, if you can get someone to keep that in their mind 1 to 2 weeks, maybe even months after he's said it, he has done his job. And he has done his job in a way that is incredibly effective. So that is more important than all of those other wonderful details that go into his research that later on you might want to explain to someone who's maybe interested in the project and wants to hear all of the details. But there's a time and a place for those details. And it's not when you only have a couple minutes and people are distracted and tired and, thinking about their day. So yeah, start and end with the big picture. That's what you want people to remember. So, and then again, every detail needs to reinforce the main picture. So in terms of the nervous system and aging, what would you say are the most important details?
So I think the most important 2 details are actually, I would say, there's 3, and I over detailed them, talking about a lot of gene names, talking about cell names, which I think was not necessary. I think I would emphasize the fact that I'm not looking on neurons, but I'm looking at glia, which are these support cells for neurons. And then the second would be that I am interested in mitochondrial stress, like as the cellular basis is mitochondrial stress pathways. And then the third one would be that there is some secreted signal that these glia cells, when you change their mitochondrial stress resistance, these cells secrete some signal that is actually on the basis of the longevity phenotypes that we see. So usually I would not go into any of the details, no genes. If anybody wants, they can ask me to do what are the genes that I'm using and what are the techniques and everything. But I would just, these are like the 3 points I would try to emphasize in like the middle part.
Yeah. Yeah, okay. So essentially, you're looking at how the nervous system affects aging. And when it comes down to it, you're looking at a component of the nervous system called glia. They're not the neurons, which are the big flashy players of the nervous system, but they're these cells that act as a support system. And in terms of what's going on in these cells that are supporting these neurons, you're looking at the stress response. And there's a certain stress response that's happening within these powerhouses of the cell, the mitochondria, that are providing all of the energy to keep these cells running, that if it gets affected, affects longevity. Would that be a correct assessment?
Exactly. Exactly.
Yeah. And see if you pare it down, and you're selective about those details, and you're very intentional about, okay, what are the most important things, and how do I create a logical flow of information? You can explain your work in a very, very short amount of time in a way that's going to stick with you. Like I'm going to remember, okay, like Roz, he's trying to understand, the effect that the nervous system has on aging. And he's trying to understand a mechanism that is involved in energy regulation of these support cells called the glia. So, it's short, It's simple and it's memorable, and that's what you want. So thank you so much, Roz.
Thank you for your feedback, Rachel.
Absolutely. Okay, I think we are ready for questions.
Excellent. Thank you so much. If anybody has a question that they haven't put into the chat yet, please put it there. I have some questions that we've collected from attendees and from our workshop. So question is, how do you conquer nervousness when speaking to the public? Allie or Rachel?
I love this question. So I've never stopped being nervous. I've been giving talks for a long time. The nerves for me are just part of it. And what I like to tell myself is that it means that I really care. So I'm going to do a good job. I would say practice. I probably practiced this talk. Ooh, it's all I've been doing for, like, in my mind, for 2 days, all I've done is practice this talk. When I'm walking around, I'm talking to myself. When I'm in the shower, I'm talking to myself. I practice like crazy. And that helps me know that, kind of know whatever, no matter what's going to happen during the talk, even if my slides disappeared, I'd be able to still give you this talk because I've already got it locked in. So, you know, if I'm nervous, maybe I'll have a backup. You know, I used to carry around like 2 backup drives, email myself my talk, send it to someone else. I have backups, so I'm not worried about the technology failing me. If it fails me anyways, you know what? I'll give you an elevator pitch about what's important about my research. And you just kind of live with the nerves. Don't eat anything ahead of time that's going to make your stomach hurt.
Yes, and my answer is also I haven't conquered my nervousness. Now, some of that nervousness is good. That little bit of adrenaline helps you think a little sharper, keeps you on your toes, but it's hard. And a lot of times what I'll do is I'll practice and I'll practice and I'll practice. I also find it's good to, you know, find your method of stress control. So, like for me, as someone who's always a little bit of anxious, I spend a lot of time at the gym. So, and also again practicing, because that's a way to go over it enough times that if you stumble up a little bit, or if the way you realize, you can get back on track a lot easier. You can also improvise a little bit. So,
I'll also
yes.
tell you guys, I'll add one more thing. I dressed up for this talk. I really believe in actually, like, you know, putting on the clothes that make you feel powerful and capable. To me, it makes a difference. So I'm in a room of my house wearing a blazer on Zoom, like I showered. So, you know, little things like that you can kind of convince yourself that add to your own confidence level. Whatever those boosters are, do them. If it's exercising right beforehand to get your adrenaline way up, do it. If that's a night before thing, do it. Whatever makes you feel good, build those practices in and do them.
Awesome. Thank you. Rachel, did you have something else you wanted to say? Okay. Yeah, so a question from Mel, especially in like a lightning talk. Is it distracting to say that we work on worms or fish? Is the study organism relevant for a lightning talk?
So that depends on how much time you have. If you've only got 1 minute, then you really want to focus on bigger impact, and you know, you might have one line saying, you know, we're studying this in a model system called C. elegans because, you know, it lets us easily manipulate these processes. But you should not start with, I'm working in a model organism called C. elegans. You should start with the big picture. So.
Awesome, thank you. And then I think a question a lot of us have as we begin to distill our work down into these core concepts is, how do we balance between getting all of your audience on board, making sure people still know that you're an expert, and not sounding too naive or like someone who can only give a big picture, right? Show you're an expert, but also get everyone on board. How do you balance that?
So first of all, understand your audience. If you're giving a Friday department seminar, yeah, you can go into a little bit more detail, like absolutely. If you're giving a talk to a general audience, it's okay to pare the details down and realize that, yeah, there are some people in the audience that will think this is too simple, but then there are also people in the audience who are going to be intensely grateful that you haven't thrown too much detail in them. Because it's not a fun feeling sitting at a talk thinking, I don't understand any of this. So, so yeah, know your audience, and it is okay to simplify more than you might think. And even your audience members that know a lot about this subject will probably be a little bit grateful too. So.
I love that advice, Rachel. I think it's totally on the nose. I think that a lot of times we turn to jargon because we think it makes us sound like an expert. And, you know, scientific jargon is so specific to each and every micro field. It really ends up confusing people and taking, you know, again, they'll turn off and you'll lose them for some part of your talk if you do something to turn them off. So I really do agree. Make things as simple as possible. Like I pointed out in my talk, I'm not going to tell you about genes and promoters because that's honestly, it's jargon at a certain point. And if someone wants to know, hey, what transgenic line did you use for that research? They can ask. And you can give them that information then, and that might be super helpful for them. As an individual, your whole audience doesn't care. So yeah, I think that if you're on stage, there's already an expectation that you are an expert or you're on your way to becoming an expert. So I think, you know, acknowledge to yourself that you're on stage because you're the expert and that your audience is on the same team and they want to understand what you're going to tell them. Like you're all there for the same reason. So help them out. Keep it simple.
Exactly. And one of the main things I did when I started writing for a general audience was I learned to be very, very honest with myself about when my attention was wandering. You know, like if I was reading something or if I was listening to something, okay, when was it that I started getting lost? Because having this sort of self-awareness helps you understand your audience a little bit better. So, you know, when you were reading a paper, when you are listening to a talk, start noticing those moments where your mind starts wandering a little bit, or maybe you get stuck on something. Because if you can detect these moments and you can be honest enough with yourself that, hey, like my mind's starting to wander, or like I just don't understand this, even though I think I should. That's going to help you understand your audience a lot better, and it's going to help you learn how to avoid those moments for others. So.
Excellent advice. Thank you so much. So we have actually come to the end of today's workshop. Thank you all for being with us today, and thank you to our panelists for taking time out of their busy days to share their experiences and provide their insights. I'll give them an opportunity to maybe add some closing remarks or put links into the chat for how people can get in touch with you or your work. So Allie, would you like to share how people can get in touch with you or your work? Unmute yourself, please. Sorry
about that. Yeah, get in touch with me. You can send me an email at rosenbergscientificconsulting@gmail.com. I'll put that into the chat, and also these slides will be shared. So please get in touch. And you can also find me on LinkedIn. Please connect with me. And thank you again so much to the organizers and to all of you.
Awesome. And Rachel, how about you?
So you can contact me either via email. My email is rcfairbank@gmail.com, and you can also find me through LinkedIn. I'm also on Twitter. So.
Excellent. Thank you so much. And Rachel is also a freelance writer. So if you Google her name, you'll find some really fun articles that I have enjoyed.
Yes. Very eclectic part.
So please stay tuned for our next Workshop Wednesday events. We're gonna take a break over December, but we'll be back in January. The topic is in the works, but it is centered around building effective mentoring relationships from both the perspective of a mentee and a mentor. So the Career Development Subcommittee will be putting together a resource package based on today's discussion. There were a few questions that we didn't have time to get to, so we will pester Allie and Rachel to get their answers to the questions. So follow GSA on Facebook, Twitter, and LinkedIn, and be prepared to check your emails to get the updates as soon as we distribute them. Thank you and stay safe.